Okay. Can you all hear me? Can you all hear me? Good. All right. So that works. Can you all hear me? Good. All right. So that works. Can you all hear me? Good. That works. Okay. If I want to join chat, then I can go to the chat button. No, I want to be able to chat. Cancel. Cancel. Alright, let's make it smaller. Chat. There we go. Got that. Alright, so then let's see. I want to start a photo booth so they can see my face in the corner.
Hey folks.
Hi, I'm Ram Gopal from presentationprocess.com. Please consider subscribing to this channel to get notified about new videos I post in this channel. In this video, you will learn how to change your desktop background for screencast videos. Whenever you record screencast videos, chances are that you may have to access your desktop. Maybe because you want to access certain software, or you may want to access certain file that you have stored on your desktop. So how do you set up your desktop background the right way so that your audience has a good feel about your demonstration? I'm going to give you all the files, put them into a specific folder, stop. Since I do PowerPoint tutorials and promote PowerPoint Mastery program, I have a commercial of Ram Gopal's PowerPoint Mastery program on my desktop. So when I access my desktop for any software or any file, all that the audience sees is this commercial, which still keeps them in line with the theme of my screencast demonstration. Now I'm going to show you how to create
So then let's do general sketch type things. Doesn't work, right? There we go. So here's my ribosome, right? There's the mRNA, tRNA, the amino acids, and they're going to continue to bring those things one by one. As always, trying to draw, press escape to draw pen, done, control. Sure. Word document. Okay, I'll do that now. Rad. There it is. Okay. So I have to search this out. Alright. Schedule this right. Schedule. Can't schedule it. Okay. Copy. I think I can't schedule it because I can't schedule 3 classes at once. I can schedule 1 class at a time. The 17th, that's today, at 11:45 it's 11:50 a.m. That's today, 11:50 a.m. Right? There. Okay. Okay, now I'm going to go to Bio. Let's do 2 here. Announcements. Paste. Back to you. Copy. Attach link. Paste. Current link. Go to schedule. DAM. 17th schedule. Really? Let me see. Hey, not bad, darling. It's
And
really
it looks good on both
Yeah,
sides.
it does. I'm proud of you. Doing great. Alrighty. I am just, yes, you're very cute. Copy. Yep. Mr. 3, announcement. Paste. Okay, got the link, which is here. Copy. Link. Right here. Add link. Schedule. AM. 17th, which is today. Okay. Good. Let's check that and make sure we have a schedule going out there. Okay. This is on my home computer. Okay. Log in. Okay, do we have a scheduled scheduled announcements? Today we found Google Classroom. So Classroom. Live stream. To be live stream. Schedule. Alright. To the second. Schedule. Alrighty. That means we need to go back to the classroom. Okay. Switch users. Alright. Back to this. To edit that. Post. To settings. 5. Period 5, period 4. Schedule. Okay, all done and ready to go. Is this? Participating. Chat, manage participants. Okay, that's good. I think we're ready to go. Almost ready to go. Alrighty, let's go to Gmail. Let's actually use Gmail. What was I going to do again? Ah, that's right. I was going to trial. Let's outline. Let's outline for translation. Now go to let's check okay. Just this week, adapted to this one plus. Study guide 120. That wasn't. Yeah. All right, I'll go get it. Here we are. Okay, so we got this one up. Let's download it. There we go. Let's make it bigger. Come on. There we go. Okay. Minimize. Alright, let's pull back up this. Get rid of that. Get rid of this. Get rid of that. Get rid of this. Alex, we can have minimize. Go back to there. Okay. And minimize. Alright, good. Cool. down deep for this one.
What? I'm sorry, I had my headphones on. What'd you say?
Is there anything you want me to sew or repair?
Yeah, there's a button on my work jacket that came off.
Not sure I want to exit, actually. In fact, I know I don't want to exit, so don't be a poop. And the stream says it's still going on. sand this first. Sand this down, make it smooth. Leave it around for a moment. Okay. Still into demonstration.
Bless you.
hello person, whoever you are. I can't see who it is, but Tom Brady. Cute. Hey Nick, how's it going? So we're going to get started here in a bit. We'll wait until some more people get on, but in the meantime, if you have a question, you can go ahead and ask, and I'll do the best I can to explain it. Also, can you hear me? Great. There's a little bit of delay, some lag between you and me. It's normal, so don't feel like I'm ignoring you. I just haven't seen what you've asked yet. Alrighty, 5 people. Well, let's get started. Why not? Okay, there we are. If you guys would do me a favor when you get into the stream, if you could just say who you are so I know who's here. And then I'll just leave it up to you guys. We still have another 10 minutes before it officially starts, but I'll just start with the first question if you guys want to begin. Hello, Ethan. And once again, I just want to double check for all you newbies who just showed up, if you can hear me well enough. Let me know. Max, hello. I don't know if this is something that will... Six current viewers. Doesn't tell me who you are. Okay. 6 current viewers. This is fine. This is fine. Alrighty. So we got 6 people. Again, I'll leave it up to you. Whoever wants to just start with the first question, I'll just begin with that. Okay. We got another few minutes before we officially start, but again, if you guys have questions, go ahead. What's the best way to draw DNA? Alright, so to draw DNA, if I asked you to draw it on the test, which I won't, I would honestly just draw like a ladder. I wouldn't even ask you to really twist it. I would just have you draw 2 lines down the middle, if I can demonstrate it, I guess. Let me go to OmniDazzle here real quick, and I can make the pen width a little bigger for this. Let's do that. If I were to draw DNA, I would probably, do one side, do another side, and then, base, 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 base. I would say this is good enough for what I would expect from you guys. If you see what I'm doing here. Like that. Cool? If you did get a picture of this, though, I would ask you to, the picture I provide you, if I do, will be much more detailed than this, obviously. And I would recommend you make sure that you can identify the parts of DNA. So you know that, you can't see it here, but there's a pentose sugar, phosphate, attached to a pentose sugar, attached to a phosphate, right? You know that these different colored lines are your nitrogen bases. So your deoxyribose sugar, your phosphate group, those going up and down the sides, and your nitrogen bases linking in the middle, right? So Ethan, I'll start. I'll tell you a little bit about the test. It's going to be mostly, or a big chunk of it, multiple choice. There's going to be a matching section, meaning there will be some key vocab words, like think of some of the enzymes that we've learned about in this unit. Some of that type of vocabulary, and as well as descriptions, you're going to have to link one to the other. The different processes as well, transcription, translation, replication, make sure you know what's happening differently between each one, what's made, and what you started with, what you end with, as well as where they're happening. Make sure the other sections are going to be, your typical questions, like what you've seen on tests in the past. For instance, look at this picture, tell me what's going on, what if I did this, how would that change that? And just make sure that you can identify what process we've studied, whether it's replication, transcription, or translation, if I were to give you a picture of that. So I would again strongly recommend you guys look at the study guides. They have some fantastic images that will kind of help you train yourself to identify what it is you're looking at. All right, next question. I'm sure you guys didn't come just to stare at my face. Is there going to be a binder check? Good question, Nick. Normally I'd say yes. But since it's going to be such short blocks tomorrow because of the assembly and the chorus presentation, 50 minutes isn't enough time for me to really get into your binders and make sure everything's there that I want to see there. So I'm going to do binder checks over the next few days, just during class while you guys are working on stuff. I'm going to just ask to see your binder and kind of plug it in as we go. And once again, I apologize for any sort of delay between your questions and my responses. There's a little bit of lag. So what I'd recommend you guys doing is practice by writing a paragraph, if you have the time and the opportunity. What we kind of did as the do now in class on yesterday, where you filled in the blanks. See if you can't write a paragraph from A to Z saying, okay, we begin with transcription, that's when this blank gets copied into this blank, then the next step is when that thing that was made leaves and enters the process of translation. I would try to organize your thoughts and make sure you know what's happening each step of the way. Would it be helpful if I posted some videos to review in the classroom? Okay. I'll post a couple of animations, some of the ones that we've seen, as well as any others that I find that are, you know, of good quality. I'll post those to the classroom for you guys to watch if you want. But there's tons of them out there. They're all pretty good. But if you don't know what to look for, I'll try to filter out the good ones from the okay ones and post them there. So any other, since it's officially now 12, is there any other specific questions since we're beginning? For instance, you know, what's the sequence of steps in transcription and translation? What do you make in translation versus transcription? How does DNA replication happen? So I'd recommend, if you guys have your study guides available, kind of pull them out and look through the questions and just ask me. I also have the study guide digitally here, so I can pull that up as reference. If you say number 8 or the question on replication, I don't really get that. I'd be happy to kind of go over that with you. Our assembly, let me pull that up, Nick, just a second here. I think that's a recent email I got here. No, that's my personal email. You don't need to see that. Let's move that over a little bit. Oop. Nope, come on. Okay, there we go. All right, let's go here and find that email. I'm pretty sure it is this one right here. No, it's down here a little bit. There it is, Thursday, day 4. All right, so it's going to be during the advisory, Nick. If you see this here, it shows you your schedule. So it's a super advisory. It's a day 4 to begin. The advisory is 88 minutes long. It's so that you guys can see the choral presentation. Hey, Burba. So it's 4 or 5 advisory. You're going to begin your advisory at your class where you normally have advisory, and then halfway through you will either start by going to the assembly, or halfway through you'll switch with the kids there and go to your assembly. So the classes are only 49 minutes long each, you know, approximately. So they'll be pretty quick. So the test has been, you know, kind of edited down so that it can go, so that it won't take as long as a normal class period, if that answers your question. So once again, there's the schedule for that. Okay. All right, good stuff. So then going back to this. All right, so the first step with transcription, can I get a refresh of the steps? Yeah, so we can do that, sure. Let's see if I can't sketch it out for you guys. All right, let's move that there so we can see what's going on. All right, so if you remember, let me do this. Inside of a cell, here's our cell, right? You've got this thing called a nucleus. There's our nucleus, right? In the nucleus is the DNA double helix. There it is, right? Okay. So here's our nucleus. Now transcription happens first, and it has to happen in the nucleus, because in order to do transcription, you need to be able to work with the DNA, right? So DNA is only ever found in the nucleus of eukaryotes, so therefore transcription happens in the nucleus. Now the first step, if you recall, remember the cookbook analogy. This is a giant cookbook. It's got 25,000 genes approximately that code for human beings, if it's one of your cells, right? So that's 25,000 different proteins. During the course of a day, or at any period of time, the cell needs certain proteins to do certain things, whether it's proteins to break down food for energy, or proteins used for growth or for signaling communication between different cells. These, or even the proteins, if it's a red blood cell, that help it transport oxygen. But these proteins, the recipes to make them, are found in this nucleus. but the proteins themselves aren't made in the nucleus, they're made out here. So if you remember our kitchen analogy, there are these ribosomes out here that are responsible for actually making the proteins. But they can't go in here, they have to get something to go in and come out, or to be made here and then come out to get to them. So transcription is how you do that. If you remember the analogy, the ribosome is like the chef, and this RNA polymerase is an enzyme that will unzip this DNA strand in order to make the mRNA strand. So I'll show you real quick. Let's see if I can do that. All right, so if I have a strand of DNA, let's kind of blow it up here a little bit. Let's make it bigger. Here's my strand of DNA, right? And let's say it has been unzipped. This bulge here is the unzipping part, right? So you've got your bases on either side. Imagine there's, bases. These are all A's, T's, G's, and C's. Even though they're all the same color, imagine that they're all different bases. Okay, and you've got bases here. Let's say the gene is on this side. Remember, AUG, or if this is DNA, TAC, is where the gene starts, right? So TAC, that's how the RNA polymerase generally is like, okay, this is where I begin. And the RNA polymerase is going to, let's make that a different color. How about we make it green? Yeah, so the RNA polymerase is going to go down the DNA strand, base by base, and it's going to attach bases of mRNA to the DNA strand to match the gene. There's that, right? And the RNA polymerase is going to work its way down the gene, copying the strand of mRNA until it's complete. And that strand of mRNA eventually, once it's done, will leave. We've got our wibbly wobbly strand of mRNA now, right? So remember, the M stands for messenger RNA because this is a coded message, right? This here is a message for how to make a protein. Our mRNA strand right here, mRNA, all right? So right now this is all in the nucleus. We've kind of zoomed into that nucleus right here, okay? So the RNA polymerase, this kind of green blob, creates the mRNA strand by reading the DNA strand for that specific gene. It'll make that mRNA strand. That mRNA strand will leave the nucleus. So if we go back and redraw our cell with our nucleus and our DNA, and there's our nucleus and there's our DNA strand, right? Now we've got this mRNA strand that was made here, and it's left the nucleus. This is transcription that we just did, right? So here's my mRNA strand, that green mRNA strand. It was made here, and it's left. That whole process that just happened in there, that was transcription, right? We've made our message, and that coded message now is going to go on to step 2, which is translation, right? So this right here would be the ribosome, because ribosomes are what make proteins from the mRNA message. Sorry about my handwriting, I'm using a mouse to do this. Okay. All right. So, Nick, that's transcription right there for you in a nutshell, right? Now, here's what you need to remember. The central dogma of biology is that DNA, right here, codes for proteins, right? DNA gets converted into RNA in transcription, and then the ribosome transcribes that message into a folded shape, right? It's got all sorts of different colors, different subunits to this thing, and that is our protein, right? Protein. So the central dogma of biology, remember what we've been learning about, is that DNA is transcribed into RNA, which then gets translated into protein. Your genes code for your proteins. This protein might be keratin. It might be the protein that's going to be used to build your hair or your fingernails. It might be collagen, the protein that keeps your skin elastic, your skin cells elastic, right? It all depends on what type of cell you have and what that cell needs in the moment, right? Okay, so DNA to RNA to protein. This step is transcription. Woo. It's all over the place. Transcription. And this step right here is translation. All right. Make sense? What else? Guys, I think I can also post this video to the classroom as well. If at any point you're like, oh no, he moved too fast, I don't really get what he just said, you can also rewind the stream if you want. At any given time, you can go back and watch stuff again while this is happening. So, just so you know. On the test, you need to know the full name of the bases. Allie Brooks. I don't recognize that name, but Allie. Chances are you will not technically have to know their names, but if you don't remember their names, remember good test-taking skills, folks, which is that typically the answers to a question on a test can be found in other sections of the test. If you don't know what the name for something is, oh, Hannah Conway. Of course. Hey, Hannah. How's it going? If you don't know the name of something in a question, look around the test. Chances are, in one of those paragraphs or one of those multiple choice questions, the name for that thing you're looking for is there, right? So that's just good test-taking practice. But Hannah, me specifically, as your teacher, if you wrote A sticks to T, I wouldn't take points off, because you know A and you know T. If you didn't say specifically adenine and thymine. Hello, Patrick. All right, so for those of you just joining us, we were talking about scheduling, what the test is going to look like. I'll go over that again really quickly, just for those of you who are just getting here. Big chunk multiple choice, smaller chunk matching. Hello, Ryan. Make sure you know the key enzyme players that we discussed, right? And their roles. So a matching question might be ligase, and you would have to find the description for ligase in the matching description section, and kind of draw an arrow one to the other, or write the letter next to the definition, right? Are there any questions about the people like Watson and Crick? Unfortunately, Hannah, no. We spent a lot of time discussing that whole historical event. But since we had to move the test to Thursday, and Thursday is now a super advisory day, we had to cut the test down, and so we removed the historical questions from the test. So, no, you will not see questions on Watson and Crick on the test. So big chunk multiple choice, a matching section, sorry, and then I forgot to continue, Ryan. And then there will be, like, remember the cancer question you had on the cell cycle quiz? It had the picture of the cancer tumor next to normal cells, and it asked some questions like that. There's going to be 1 or 2 questions similar in structure to that. There will definitely be a transcription translation practice question, like what we did with the activity yesterday, right? So I might, in a question, give you a strand of DNA, and I might say, take the strand of DNA and transcribe it, right? Do what RNA polymerase does and turn it into mRNA, and then take that, and I'll give you an amino acid wheel perhaps, and I'll say, okay, now plug those codons you just transcribed into your amino acid wheel and tell me what amino acids in what sequence you have. You guys were showing me that, you know, that's something that you can do for the most part. I felt like most of you had a handle on that. The test itself is going to be, let me see, how big is that test? I think it's about 20 multiple choice. I want to say 8 matching, and then 3 short answer and 1 essay. And the essay, you know, it's not going to be like write a paragraph. It's just going to require more work than like an average short answer. Sure, Ryan. I'd be happy to. Okay. So let's do that. Other than translation and transcription, I think that's what you meant to say. What else is on the test? So before I start the chef analogy, Soleil, I'll tell you DNA replication. Remember, how do I have 1 DNA strand and turn it into 2? We learned a lot about that. You know, have to pull it open. There's an enzyme that unzips it. There's an enzyme that copies it. There's an enzyme that sticks it all back together, right? You have to know things like semi-conservative. We went over that word, I think, a number of times. Why is DNA considered, or the replication process considered semi-conservative, right? Semi meaning half, conservative or conservation meaning to save. Why is DNA replication save half? How does that work? Hopefully that jogs your memory. I imagine most of you can think of what that means. So yeah, Ryan, for the chef analogy, let me get rid of that. Oh, sorry, wrong one. There we go. Okay, so for the chef analogy, Ryan, remember you have your kitchen, or you have your restaurant here, like that, right? And in the restaurant, there's a secret back room, and there's the kitchen area. The kitchen area has a pantry, it's got shelves with ingredients on it, right? So we've got things like fruits and vegetables, there's some tomatoes, right? You've got, you know, your meats and chicken and all that, right? There's some chicken right there, right? Okay, so you've got your pantry, and you've got your chef, right? He's sitting at the, or he or she is standing at the stove, Ryan. Here's our stove with our fume hood there to get rid of all the smoke. There's our fire, okay? And inside of our secret room, you have on a pedestal, I'm just going to redraw what we did in class. Some of you are going, oh man, not this again, but some of you, I think this might be helpful. In your secret room, you've got a giant cookbook, right? And this giant cookbook, let's draw lots of pages, pretend it's got lots of these pages. It's a huge, huge, huge cookbook. Now here's the thing, this cookbook is too big to leave this room. The chef cannot go into this room and get this cookbook. First of all, the head chef is really important, and he doesn't have the time to go running back and forth. He has people doing that for him, right? Okay. So our head chef today, you know, he wants to make something. He's going to have somebody copy the single recipe that it needs in here, because this cookbook has 25,000 recipes, shapes of different types of RNA. You should, yes, you should know the different shapes, right? You should know a ribosome is a big, lumpy, knobbly looking thing. You should know that the tRNAs look like T's, and the mRNA just looks like your regular single strand, right? Okay, so you should know the 3 types of RNA and what they look like. So there are 25,000 recipes in this cookbook, right? And the chef only needs 1. So he's not going to go and take this whole cookbook out for 1 single recipe. That's a huge waste of time and energy. So he's going to send a little sous chef in to copy that for him, right? Here's our little sous chef today. He's got his fancy sous chef hat, okay? And the sous chef, his job is to copy just one recipe. That's all the chef needs, is just one recipe. So the sous chef is going to open the cookbook and copy one recipe. And we'll draw the recipe in our sous chef's hand that he's copied here. All right, there's the sous chef's recipe that he's copied here. Okay, there it is. The sous chef is our RNA polymerase, right? This is RNA polymerase. Because RNA polymerase copies the DNA. That's its job. RNA polymerase. The copied recipe today is our mRNA strand. It represents the copied gene that was copied out of this giant cookbook, which is our DNA. Right? So in your cell, your DNA has 25,000 genes on it. It's a huge cookbook, right? And those genes are spread out through 46 chromosomes. But the RNA polymerase only needs to copy one gene out of those 25,000. There's the one recipe out of the 25,000. The RNA polymerase, our sous chef here, is then going to throw the recipe in paper airplane form to our head chef. There it is, right? So the mRNA, all of this first step here, everything we just did here, this whole thing is transcription. Transcription, the first step. Okay, so this first step is transcription. DNA, as I hope all of you remember, can't leave this area, and this area represents our nucleus, right? Our nucleus. Okay, so far so good? So transcription happens in the nucleus. It's when RNA polymerase copies a section of DNA called a gene into an mRNA strand, which is then going to leave the nucleus. This, everything that just happened here, is transcription. Right? The next step is translation. That's everything that's going to happen here. This is all translation. And we can go over translation as well in a moment. Okay? So, and since this is the nucleus and this is a huge building, we're going to call this whole restaurant the cell. All right? So the RNA polymerase in transcription made a copy of the DNA. There's the copy. That mRNA strand, this happened in the nucleus, is going to leave the nucleus and find the head chef. And if you recall at this point, I'm sure many of you are rolling your eyes, oh, this analogy, man, I'm so tired of this. If you recall, this head chef is our ribosome. The ribosome receives the mRNA message from the nucleus, and it tells the ribosome, okay, what ingredients do I need? What order do they go in order to make a finished thing? Like here's our pan, right? And there's all sorts of like goodies in this pan. Ingredients are being mixed together, and this food that's being cooked represents, what does it represent? Somebody type it in. This finished product, the thing we make at the end of this whole process. What is it called? There's a little bit of lag, so I'll just hang out and wait until somebody says that. Proteins! Thank you, Soleil. That's the protein. Yes, indeed it is. There's our protein, right? Proteína in Spanish, I believe. Good job. You guys remember that. Awesome. Okay, so protein. There it is. That's the protein that's going to be made. But the chef, our ribosome, doesn't know how to make that protein until it gets the mRNA message that was created in the nucleus, right? So when the chef does get that, translation can begin. It's going to read the chef this mRNA message, this recipe, and it's going to order its little sous chefs to deliver ingredients step by step, right? There's a little sous chef. And the sous chef is going to bring plates with ingredients on them. There's an apple, we'll say. Maybe he's making a strudel, right? Okay. one by one, these sous chefs are going to grab ingredients and bring them to the chef so that he can combine them to make the meal. So this is the last part of this analogy that we need to go over, right? The sous chef, which looks like a letter of the alphabet, perhaps, a little bit, right? Okay. That sous chef, we all know, is... Any guesses? Somebody knows it. You knew protein. I bet you know that. Yay, thank you, Nick. Yes, it is tRNA. Good. Yes, it is. Okay, it must have just been the leg. That is tRNA. The protein is made of many ingredients. If you remember from trimester 1, the monomer of a protein, there are 20 different kinds, and those guys are called what? The ingredients. There's a lot of lag. I'm not going to wait around. I'm sure you're all thinking it. It is the amino acids, right? So the tRNA delivers the amino acids, good job, yes, amino acids, to the head chef, who combines them into the protein. Boom, once the protein's done, that's it. The cell can use it for whatever it needs to use it for, whether it's making fingernails or making hemoglobin or creating ATP synthase so that it can make energy for you from the food you eat, right? All of those proteins and those enzymes are important. Even the RNA polymerase, this is an enzyme. It has to get made through this process, right? Even the guys that are making these things and playing a role in this, they have to be created through this process, right? Pretty interesting to think about. Would you guys like me to kind of illustrate this? Because I already illustrated the real process of, oh, where's our RNA? Great question, Aubrey. Where's our RNA? The ribosome, the R in ribosome, or the R in rRNA, stands for ribosomal RNA. So the ribosome itself is made out of RNA somewhat, a little bit, so that it can actually read the RNA message that's found in the mRNA. So the ribosome itself is rRNA. And let's think about that for just a moment. This process is called translation. Translation, if you think of where that word is and where it's used in your everyday language, right? Translation is when you are translating one language into another, right? So if I want to translate, I don't know, Soleil is speaking French, maybe, I don't know if she actually speaks French, but Soleil is speaking French, and let's say Jose is speaking Russian. If I speak both, I can translate. I can take the Russian and I can turn it into French, or I can take the French and turn it into Russian. So the ribosome, in order to be able to speak protein or to be able to read this RNA message, it has to be part RNA and part something else to make the protein. That's the translation part. It can read this language and it can turn it into this language. That's what's going on there. Okay, so Soleil, is replication and transcription the same? No. No, they are completely different processes. They are done for very different reasons. The only similarity between replication and transcription is that they both involve opening up the DNA in order to make a copy of something. That's the only similarity they have, right? So in replication, you take your DNA strand, right? I'll draw it down here actually. Let's start over again. I'll post this whole video up on the classroom so you guys can watch it and fast forward and rewind as you see fit. But so in replication, I have a DNA strand, right? There's my DNA. It's not twisted at the moment, so there it is. You've got your bases on either side. Now if I took the time, I'd have 4 different colors for you guys to see, representing the A's, T's, G's, and C's, but I don't have the time. So you have your normal strand of DNA. In replication, that strand is going to get opened up. Right, so here's my new strand right there. And who does the unzipping in this process? Who is kind of cracking this open here? I'll give you a moment. Remember the enzyme that unzips the DNA. It's not polymerase in replication. Polymerase does this in transcription, but polymerase does not do this in replication. It's a different enzyme that does this in replication. It starts with an H. Helicase. Helicase. So the way I remember this, guys, is that what shape is DNA? It's a double helix, right? Helicase unzips the double helix. That's why it's named that, helicase, right? So helicase comes through and kind of blasts apart the hydrogen bonds that hold the two sides together, right? So helicase opens up this DNA strand by breaking the hydrogen bonds that are formed between the bases, okay? So here he goes, he's going down the zipper, blasting apart the hydrogen bonds, and you can see the DNA opening up above him, right? Here's helicase. Helicase. Right? Okay, there's helicase. The next step, if we get out of that real quick, the next step is that after the DNA has been unzipped, like this, the next enzyme comes along and starts adding bases. This guy's going this way. Remember, because DNA is anti-parallel. And this guy's going this way. And he's adding new bases onto the old bases, right? What are these green guys called? Helicase unzipped it. Who are the builders who are coming along and building the new strands? What's their name? That's the polymerase delay, yes. Okay, so polymerase builds, helicase unzips. The difference is in transcription, RNA polymerase does the job of helicase and its normal job, right? So this is DNA polymerase. Okay, next step, let's move it down here. After you've created your two strands, these polymerases have gone by and done their jobs, right? You've got boom, 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 and yada, 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 yada. Same thing over here. I'm sorry for my childlike drawing skills, but using a mouse is not the easiest thing. And, oh, no, wrong one. And the polymerases are gone. They've done their job. They've built their bases, added their bases onto the strands. There's one last enzyme that's going to come along and tie these things all together, meaning it's going to kind of connect all of the backs together, right? And that's the ligase. That's the gluer. Remember, we learned about ligase. Close, Aubrey. Ligase, right? Ligase, these guys, after the bases have been attached to the originals, they come along and secure everything and make sure all of the kind of backbones are secured so that the stuff doesn't just fall apart again, right? So that's your ligase. So we've got helicase, polymerase, here's polymerase, DNA polymerase, right? You've got RNA polymerase and DNA polymerase. DNA polymerase speaks in T's, RNA polymerase speaks in U's. And then here you've got ligase coming up and gluing, all right? Right there, okay? And now look, at the end, you have boom, these strands. Yes, you're right. I didn't include those. Hannah, you're absolutely correct. Primase also does tell the polymerase where to start. I didn't include those because it's not an enzyme I'm going to test you guys on. I really want you to, again, polymerase is really the biggest one. If you know helicase and ligase, that's awesome, but polymerase, if you remember nothing else from this unit in biology, like 10 years down the road, if you're like, oh yeah, I remember high school biology, you should remember polymerase. It's a very important enzyme, okay? So when we're done, look, we started with this, we went through the steps, and look at what we have now. We have 2 strands, but notice how they are not, technically, if you look, even though they might have the same letters in the same places, this one's a different color because it represents the new side. So DNA, the process of replication, is semi-conservative, right? Semi-conservative. It's a semi-conservative process. Semi meaning half, to conserve meaning to save. Notice how we saved both sides of the original strand. They were saved. They didn't disappear. They weren't destroyed. They were saved. We used half, so we saved half. The other half of these new strands is new DNA, right? Make sense? Okay. So this is replication, Soleil, right here from start to finish. It's when one DNA strand over the course of this process gets turned into 2 new strands. Now, can anybody off the top of their heads tell me, why do cells bother doing this? This looks bulky and cumbersome and a pain in the neck, and there's a lot that can go wrong. Why do your cells do this, every single one of them, during the course of their lifetimes, except for maybe a choice few? What's the point of taking one and turning it into 2? Cells die. Yeah, they sure do, but why do I need to make a copy of my DNA if I'm dying? That's, you know, that is true, they all die. Keep going with that, Soleil. They do die, so we need them. What do we need them to do? Imagine if all of your cells are dying, and that's it. Yes, there you go, Aubrey. To replace your dead cells. Your cells, Soleil, do die. Most of them do, right? Except for, again, some few that last a really long time, but eventually they also will die. So if all of your cells are dying, and you're not replacing them with new cells, you're just basically decaying. Oh, no, it does say offline, you're right. Why did it go offline? Oh, okay, it says it's live again. Okay. Alright, it says it's live on my screen as well, so I think whatever happened, it's been resolved, I believe. Sorry, it must be my interwebs. Okay. I only have 2 of you now. Oh, everybody else left. Man. Okay, well, we still have technically 20 minutes. Oh, okay. Soleil, looks like maybe I'm wrong. Maybe there's more than 2 and I just don't see it. Okay, Nick's back. Everybody's back. That's good. Would you guys like me to go over translation the way I have with transcription and replication? So replication is its own thing. And I'll go over all 3 of those, Soleil. I'll do like a little flowchart for you guys at the end of this. But let me do translation first, like a little sketch up here. And then I'll kind of make a chart for you to help you organize those thoughts. So that was replication. So in translation, this is happening outside the nucleus, right? Because transcription happened in the nucleus, so if we're looking at a big old cell, the nucleus is here, that's where the DNA is kept safe, right? Transcription made a strand of mRNA which came from the nucleus, and it's on its way out to a ribosome. Now ribosomes are much, much smaller than this. Compared to the nucleus, they're invisible because of how small they are, right? And the ribosome, we'll call rRNA for short today, right? And this is mRNA, which was made in transcription. And this is DNA over here, right? Okay, the ribosome, what's going to happen over here is translation. So transcription was here, translation is here. Okay, so now let's zoom in on the ribosome and go over what's actually happening here. All right, so I have a ribosome. Ribosomes have two parts to them. They have a subunit and a major unit, or I think it's called a major unit, one of those two things, right? It kind of looks like a cheeseburger with a huge top bun. The mRNA that was made in transcription is going to feed into the ribosome like this. And the ribosome is going to read that mRNA strand. So remember, this was just made in transcription, this mRNA strand. Right? So this is my mRNA. And this is my ribosome. The ribosome is reading the mRNA strand, and here's how it knows what to do. If I were to draw a tRNA molecule. Let's say here's a tRNA molecule floating around out here. It's a T, looks like a T kind of. All right, if you see it on the test, remember if it looks like a T, it's probably tRNA. The tRNA molecules, they carry, just like the sous chefs, ingredients with them. These ingredients are the amino acids. So this is tRNA, and this here is my amino acid. We'll abbreviate that just AA for short. Okay? The tRNA is going to bring its amino acid to the ribosome, saying, hey, here I am. I brought my amino acid with me, and I know it's the right one because my anticodon matches your codon, right? And it will drop off its amino acid here when it matches. So when these guys meet, oh, boom, they stick together, those codons, the anticodon and the codon. When they do, the tRNA can drop off its amino acid right there. Now, the ribosome will move over to the next one. This tRNA is going to leave after it's dropped off its amino acid. And then the next one will do the same thing. And every time an amino acid gets dropped off, it sticks to the one that was already there. So you can't see this. No, you do not need to know anticodon. You do need to know what a codon is. That is a word you should definitely know. So every time a tRNA comes and sticks to the codon, it drops off an amino acid. And as they get dropped off, they stick together, and you can see this chain of amino acids starting to emerge from our ribosome. This chain will grow and grow and grow until the ribosome reaches the stop codon in the mRNA strand. And that tells the ribosome there's no more, this is the end. Whatever last amino acid there is, that's it. And that last amino acid will get stuck onto the chain. That chain will detach from the ribosome. And that bead that you've made is a polypeptide. Remember, polypeptides are proteins that haven't folded yet. The anticodon is the one that the tRNA... Yes, so the anticodon here is what the tRNA uses to make sure it's stuck to the right codon of the mRNA strand. Now that sounds really complicated, but it's basically like saying this has to be a mirror for this, right? If you held a mirror up here, A to U, G to C, C to G, the anticodon of the tRNA has to be able to stick to the codon of the mRNA. That's how the ribosome always gets this right. The ribosome can't screw up because these guys can't stick unless they have matching bases. Yes, so every 3 things you're seeing here, Soleil, is a codon. Every tRNA has its own little like pitchfork at the bottom of it, and that's its anticodon. The anticodon has to match the codon in order for the amino acid to get dropped off. So far, so good? So this chain of polypeptides, or this polypeptide chain of amino acids, is going to, after it's made, kind of fold up into a complicated shape, right? They'll imagine this is a 3D shape. All of the amino acids have kind of bunched up, and that's our protein, right? Right there. Now, this isn't a protein yet. This is a polypeptide. It doesn't become a protein until it's folded. So polypeptides fold to become proteins like that. Okay? Make sense? And this is translation. There, that's translation. Okay. All right, any other questions about translation? Before we move on, I'm going to give you guys a situation here, and I want you to think about what it would cause. So, to do this, I'm going to need amino acid wheel. So let me pull my amino acid wheel up. I'm sure I can find one in the Googles. Amino acid wheel. Images. There's one right there. Sure. You can use that. Let's pull it down here. Let's move that. Let's make this big. Come on. You can do it. There we go. Alright, so here's our amino acid wheel. We'll leave that right there. Cysteine, SOP, tyrosine, you can't, they're a little blurry, but you can see how we use this, right? Okay. So I'm going to give you a situation now to think about. I'm going to get rid of this real quick. Okay. So imagine that I have a strand of DNA, and the strand, actually, you know what would be better than this right here if I actually just wrote it out? Let's say it's AT, well, hold on, where's this going on? Let's make it smaller. AT, just a second here. I'm going to make this black, yeah. What's going on here? Why is this not working? Oh, that's why. Okay. Oh, that's why. Okay. So let's say it's, here we go, TAC, GCC, CAT, AGA. Okay, so here's our DNA strand, right? Now, if we wanted to create an mRNA strand through transcription, here's our DNA strand. We want to transcribe this now and turn it into a strand of mRNA. So let's make mRNA red. TAC, T sticks to A sticks to U, C sticks to G. That's our first codon. G sticks to C sticks to G, C sticks to G. Our second codon, C to G, A to U, T to A, our third codon, and U to A, G to C, A to U. There's our fourth codon. So here's our mRNA strand. Now we want to plug this in to a ribosome to make our protein. So this is transcription that happens in the nucleus. Now let's do translation in a ribosome. AUG, if we use our amino acid wheel, A to U to G gives us MET. Remember, these guys always start with MET. So let's make this protein green here, MET. That's our first one. CGG, C to G is arginine. So let's write ARG. All right, this is the ribosome doing translation now. It's taking these amino acids, reading the mRNA strand to put them in the right order. G to C, or GUA. So GUA gives us valine, I think. Let's write valine here. And then UCU gives us U to C to U, gives us serine. It looks like S-E-R. Okay, so there's our amino acid. And let's just put a stop in here because that's what it's supposed to be. Okay. So here's our protein chain, our polypeptide, right? Now, I want you to think about this situation. I'm going to ask you guys to do this for me now, actually. So whatever you have at your desk, if you have a piece of paper, yes, these are the amino acids. These are all abbreviated amino acids. There are 20 different types, but this is our gene today. It's short, right? So I want you to do this on a piece of paper by your computer or your phone, whatever it is you're using. Now, imagine DNA polymerase, when it's replicating this DNA strand way back when, it goofs up and it makes this, let me see, GCC, yeah, it makes this C a G. Whoops, that's not right. That causes the mRNA strand, when it gets transcribed, to be CCG. Now tell me what happened to our amino acids. Go ahead. I want all you guys to do this. Trust me. Trust me. Hint, hint, nudge, nudge. This is good practice. Actually, this should have been alanine. That's alanine up there. So that's what it was originally. It changed the amino acid, GCC, or GGC to CCG. Originally it was alanine. What did I do? CCG. Okay, so Hannah, here's how you need to reword what you just said. The amino acid will change leading to a mutation. A mutation happened leading to a change in the amino acids. That's what you should say. The amino acids didn't cause the mutation. The amino acid change didn't cause the mutation. The mutation caused the amino acid sequence to change, right? Now, let's think about this. Originally, this guy was alanine. What is it now? CCG. What did it get turned into? Look at that amino acid wheel and tell me what you think. This was it before the mutation. Proline. Good, Soleil. Yes, correct, Soleil. The new amino acid is proline. Okay? Whoops. There you go. A different amino acid. Now, here's why this is a big deal. I'm going to show you guys an example. So let's say you've got your original one here. You've got met to alanine to valine to serine to the stop codon, right? Now, different amino acids, just like any chemical, have different characteristics to them. So, for instance, alanine might have a positive charge. There's alanine with its positive charge, right? Valine might have a negative charge. What do we know about positively charged things and negatively charged things? And if I draw these guys as their beaded form, they join. They like to stick together, Soleil. You're right. So the normal healthy protein, this guy, since he has a positive charge, and this guy has a negative charge, when this thing folds, because remember, a polypeptide isn't a protein yet, they fold because of the nature of the amino acids, it would look like, at least between the two in question, they would get right up on top of each other, right? Because they are attracted, positive and negative, right? But let's pretend that proline, this change here, this new amino acid has a negative charge. What does that do to our polypeptide? How do negative things react to other negative things? They don't like each other. They repel each other. You're right, Soleil, right? So that means that in this case, if this were the case, the alternative, because of the mutation, these beads, these amino acids, are going to want to get as far away from each other as they possibly can because they repel each other because of their charge, right? The normal protein should look like this, but our mutated protein looks like this instead. And if you remember, a protein, its job depends on its shape. If I have a protein that's supposed to let sodium or potassium through my cell membrane, if it doesn't have the right shape, it won't be able to do that, which means that that mutation has caused a genetic disorder. So we're going to be learning about genetics next unit. This right here is what causes all of those genetic disorders that we're going to study next unit, right? So this is where all of this plays into what we're learning about. These mutations can be a big deal. Now, if I gave you a different example, if I said GGC was the original one, or let's say we change this back to GCC, or C, yeah, let's change it back to CGC, which is CGC, that gets us to alanine. So we're back to the correct sequence, right? That gives us this correct one. Now, what happens if I change this one to an A? And now we've got GCU. What happened? We'll make this the last one, I promise, and then I'll do a brief touch-up and we'll end for this session. And then we'll do another one later if you want. Take a look at the amino acid wheel. What do I have now? Normally it's this one, but I changed the code, and this is the new amino acid. It's alanine. It's the same thing, right? So Hannah, you're right. It's hard to see, but this is alanine. So I had a mutation, didn't I? I changed something, but did it have an effect? No, you're right, it didn't. This is called a silent mutation. Most mutations are silent, meaning that they don't have an effect. They don't change anything in the organism. It's only when you get a new amino acid that bad stuff happens. And I don't mean to say it's always bad stuff. It can be an improvement. Sometimes you do get an improvement because of this, but those are extraordinarily rare. And those mutations that are an improvement, if they're beneficial to the organism, can lead to evolution. Those new genes, those new proteins, might make that organism better at surviving than other organisms of the same species. And so it will cause the drive of that species evolution to have that trait in the gene pool. All right, so we're just at about 1 o'clock. If you guys have any other questions, I guess, you can, I wanted to do just real quick review with you to kind of show you what's going on. So we learned about DNA replication. We learned about transcription. We learned about translation. Okay? For DNA replication, we need to know the start. What did you start with? Let's move these over here. That's fine. Okay, let's make these just a little smaller. Maybe 10. Size 10 should do it. Right? What did we start with? What do we end with? In DNA replication, we start with a 1 DNA strand, right? Let's move these over a little bit. Start with 1 DNA strand. In transcription, we start with, let me move that over a little bit, there we go. In transcription, we started with DNA, and in translation, we started with mRNA, right? What did we end with for each of these examples? Well, after DNA replication, we had 2 DNA strands, right? Through the process of replication, we turned this 1 DNA strand into 2 DNA strands. Through the process of transcription, We started with DNA, and we turned that DNA through transcription. We made a single strand of mRNA, right? Transcription took the DNA, copied a gene into mRNA. In translation, we start with this mRNA, right? So the mRNA goes into translation. And translation, what did I do? Escape. No, DNA, mRNA, right? And translation turned that mRNA into our finished protein. Yeah. All right, fine. Our finished protein. Okay. There's translation. Let's make that one blue. Sure. Okay. So these are what you have to know. DNA replication, you start with the DNA strand, you do replication, you end with 2. Transcription, you start with DNA, you do transcription, RNA polymerase makes an mRNA message, right? You go on to translation, the ribosome uses that mRNA strand, it reads it and turns the code into a sequence of amino acids that makes the proteins. Soleil, correct. That is the big picture. Mutations change the amino acids, which change the protein shape, which change the function, right? If I normally make hammers and the recipe's wrong for how to make hammers and I end up with something that's got a metal spike rather than a hammerhead, I've made a drill. And a drill can't do a hammer's job, right? So that's, in essence, what happened there. So just remember, these are the key things for each of these processes that we've learned about. You've learned how transcription happens, how translation happens, what goes in, what comes out, where they happen, right? This is big picture stuff here. All right, any last questions before we end for the session? Last thoughts? No thanks. Fine, Soleil. I didn't want to teach you anyways. Replication in prokaryotes. Nope, you are not going to be tested on that. We, as a cohort, me and the other biology teachers decided that that is not something you guys need to know. You did not learn that. That will not be on the test. Yes, correct. What should I put on study guide? What should I put on the study guide? The answers to your questions. I don't know. What do you mean, Hannah? Oh, what should I put on? What should you study? I mean, we kind of went over it. I just kind of reviewed with you guys what everything that we reviewed just now is. You should expect to see on the test in some form. Oh, ignore those. Oh, for the study guide questions that we didn't go over, just cross them out. Cross them out. I'm sorry. I should have told you to nix those in class, but yeah, no, we're not learning about that. Yep, there is a question on DNA replication in prokaryotes. Cross it out. You're very welcome, Hannah. Soleil, thank you for coming and asking good questions and participating. I really appreciate it. This is a new thing for me, and How do you guys feel, real quick, if you could just take a minute, how do you feel this went as far as an option if there's a snow day, or maybe before every test I give you guys this option to be here and do something like this for you? Did you find it helpful? Yeah? Okay, great. I'll try to build it into what we do normally. Yeah, you don't have to call out the answers, which is great. I found that, yeah, people were participating that don't normally in class as readily, and that's good. I'm glad I can provide you a space to do that. Alrighty, well then that sums up our first meeting. Yeah, it is better than Googling and getting it wrong. I'm going to, right now, I promise you, post video clips to the Google Classroom for you guys to use if you want a visual. That's not me scribbling like a second grader on Microsoft Word. I would recommend taking a look at those, and they will greatly help you. Okay, sound good? If I see you tonight between the hours of 8 and 9, fantastic. If not, then I'll see you tomorrow for your test. Study hard, and I'm proud of you. Thank you for showing up. You guys care, and I appreciate that. Alrighty. Take it easy. Am I still streaming? Did we go offline? No, where's we stop? Exit? Oh, there we go. Stream recording will be shut down. Are you sure you